are Group E, and we investigated the bacteria found in the GT Community Garden. Here's an overview of our presentation. And so, someone said in our last presentation that over 95% of the bacteria in soil still remains uncultivated, undiscovered. And among all of the bacteria found in soil, most of the portions of it are going to be beneficial bacteria that do things like convert nitrogen and suppress pathogens, but another portion of it might be pathogens that are harmful to other plants and humans, of course. And especially because we do our bacteria from a compost bin, it's going to consist of materials that humans dispose of, might consist of more harmful bacteria. But it's important that we learn what genes produce what phenotypes so that we understand which genes may need to be up or down regulated to make sure that there is a healthy ecosystem and plant growth. So we, again, sampled from a compost bin, and it was specifically from the third bin. So it consists of very decomposed compost and food scraps. So it's almost fully soil, and it was warm air, it was dry. So we thought that would be a good, there would be a good abundance of microbes.
And then looking at our 16S results, there was 143 unique species and then 3 repeated family names. So those function as ammonia oxidizers, function in breaking down organic materials, as well as releasing insoluble phosphorus. So just like typical soil bacteria things, but not something that we necessarily wanted to study. So we ended up choosing bacteria from like the smaller categories. And then looking at our group PCR results, as you can see, all of our controls performed really well, but we really didn't get good results, each of us. These 3 have kind of like faint streaks and like separated lines, but there really wasn't like successful PCR results. That just may be due to human error or not the correct conditions, but yeah.
Okay, so for our individual isolates, I isolated Bacillus thurinensis. It's a bacillite or a rod-shaped bacteria within the Bacilliaceae family. And I chose it because there was an off-white yellow color on the petri dish, so it was very similar to the other colonies from the main petri dish from our soil sample. So I thought I'd get sort of a maybe representative bacteria that's commonly found, and I was right in that regard. It was gram-positive, as you can see from the violet color in the gram stain, and there were 19 different contigs and 6.1 megabase pairs, so fairly continuous positive, but there were definitely some pieces missing. But overall, this bacteria functions as a probiotic for animals, but it also helps plant growth in multiple ways, one of them being growth under salt stress. So some genes that I found were related to cellulose binding, flagella, antibiotic resistance, but there were 7 total genes for homolysin, and 4 of them were in a cluster. And these 4 coded for different components of the hemolytic interleukin protein, hemolysin protein. So the components were L2, L1, HBLV, and the binding component. And the binding component is the one that I chose to focus on. So each of these components, they're kind of squished together, but the 4 are here, and my focus, the binding component, is the one that actually binds to the membrane, and it forms a pore in the membrane, which can lead to ion leakage and eventual cell death. So like other hemolysin proteins, HBl lyses exclusively eukaryotic cells, and that includes red blood cells, so I decided to test it on a blood agar plate. In the structure, it matches that of literature. So the structure from literature consists of these alpha helices forming barrels, and there's high confidence in both of those regions, but for whatever reason, my protein consisted of a intrinsically disordered protein and also a beta sheet, so I thought that was super interesting. I wanted to see what makes that unique in the future. And for subcellular localization, it's found near the membrane, which makes sense because it needs to latch onto the membrane to form that pore and cause lysis. So based on this data, because the structure of my protein is consistent with that found in literature, it's found near the membrane, I predicted that it would do either incomplete or complete hemolysis, also known as alpha or beta hemolysis, on a blood agar plate. And so some phenotypes that I found was that it did do complete hemolysis. You can see in the discoloration here, and there's a zone of clearing. And then for those other genes that I had, like related to flagella, there was a fair amount of motility, is slightly larger than the positive control. control, and then there's some antibiotic resistance linked to penicillin and ampicillin, so I found genes for that. And overall, those results supported my hypothesis, and the bacteria as a whole can lyse the red blood cells, but then again, I did have 3 other genes for other hemolysin proteins, and my isolated gene was just one component of that HBL protein. So I think future experiments, I would definitely want to try knocking out maybe different parts of the other components of the HBL protein to see if it still does at least incomplete or like partial hemolysis. And then I would also want to do another experiment where I knock out all but one hemolysin gene coding for the protein to see if it still does any degree of hemolysis.
Alright, so for my isolate, similar to Shaylee, I also isolated Bacillus thuringiensis. It's a gram-positive bacteria. According to the image, you can see it's dark purple stain. A rod-shaped bacterium from the Bacilliaceae family. Its function is mostly as a probiotic in animal feed. That's how it's used commercially mostly. And some of its conditions, it's aerobic and grows at low temperatures. The cells themselves form chain links, as you can see in the image, and the colonies themselves are like a white cream color. I also chose this species, or this isolate, because I felt like it was pretty representative of our sample, and it was. So, looking at the genome a little bit more closely, it had a genome size of 2.6 megabase pairs, and it had about 305 contiguous sequences, so it was a very highly fragmented genome. But when looking more closely into the annotated genes themselves, I found a lot of genes related to antimicrobial properties, about 9 of them related to bacteriocins mostly, some chain linking genes related to things like adhesion and protein forming, some hemolysin genes. I also found some genes with antibacterial or antibiotic resistance, as well as motility as well. So looking at the bioinformatics, the specific gene that I chose was called the heterocycloinducin sonorensin bacteriocin gene. It doesn't have a more specific or a preferred name, so I'll probably just call it the sonorensin gene, but it was not part of a specific gene cluster. There were other genes within the antimicrobial genes that I was looking at that were, but this one itself was not. So sonorensin itself is a more newly discovered class of bacteriocin or antimicrobial peptide. And it's been found to be very broad spectrum in its use, and has been even used as a food preservative. A bacteriocin is basically just a ribosomally synthesized compound by prokaryotes that targets other microbes, and usually, in most cases, it does target bacteria species similar to the producer, but not always. And bacteriocins themselves are produced usually first as an inactive peptide, and then around the maturation phase, the N-terminal is cleaved off, and then exported by an ABC transporter, as you see in this image here. So that's kind of how it functions, and that's what my gene is related to. When looking a little bit more into the protein structure itself, I had a very low confidence structure. It was a pretty short sequence, and so I couldn't really gather too much from it, but it was similar to some other structures that I'd seen for bacteriocin. When looking at the subcellular localization though, however, it had an extracellular localization. This image isn't for my specific bacteria, but it kind of showcases how the extracellular localization works, which makes sense since bacteriocins are produced as an AMP and released outside of the cell to target other microbes. So with all this information, I was able to hypothesize that based on the genome analysis and function of sonorensin, my isolated species would show a zone of clearance or inhibition when growing with other species of bacteria. Looking at the results from my experiments, this is the motility assay, and I had pretty high motility when compared to the positive control. Lactose fermentation or metabolization, it was very clear, so it did not metabolize lactose really at all. Looking at antibiotic resistance, mine was resistant to penicillin and ampicillin pretty highly, but had very low resistance to tetracycline. And then lastly, the antimicrobial properties, which is what's important for my hypothesis. Unfortunately, against all 6 of the species that we tested, there was no inhibition, so there was really no antimicrobial property detected for my species against at least these 6 species. So what does this mean as far as my results go? The hypothesis overall is unsupported. There was no zone clearance, so the gene that I was looking at unfortunately likely was not producing the bacteriocins or the function that I was looking for. Some reasons for this could be that I just didn't test the ideal species. Out of the 6, I might need to test a different set of species that it might be more effective against. The gene could also just not be functioning very well or regulatory, and there also could be experimental errors that led to these results. In the future, I would definitely want to test a wider variety of bacterial species, look into some other genes to more strongly see if there is antimicrobial production really in my species, and potentially even doing some gene modification as well.
Alright, so my bacteria was Christia megaterium. I chose this because it was yellow, medium to large size, and the safe option between the two bacteria that I chose off the plate. Once I looked at it under a microscope, I noted that it was gram-positive due to the purple color, as well as rod-shaped, and that it formed chains and clusters, and it was really large. This picture is only under 40x, which I was super excited about. And then looking at the genome analysis, there was 5.5 megabase pairs and 102 contigs. And there was genes for a whole sort of things like antibiotic resistance, lactose fermentation, hemolysis, motility, and among other things as well. Looking at bioinformatic analysis, I know this is a wordy slide, but I'll explain all of it. So there was 5 gene loci. Actually, I'll go ahead and say I chose hemolysis as the genotype I wanted to study, and there was 5 gene loci identified for hemolysis. Within those 5 gene loci, there was 2 different types of hemolysis genes. So I chose hemolysin 3, and I identified no clusters for hemolysin 3 within the genome, and that the subcellular location was the plasma membrane, which makes sense because it allows for lysis, as Shaylee has already discussed. And then looking at literature, I found that there's 2 functional domains. That's for membrane binding and pore formation. And to explain a little bit more about pore formation, I'll go into the transmembrane topology. So basically, within hemolysin 3, cells that have this protein bind to the target cell membrane. And after they bind, they form like a ring-shaped complex. And then there's some folding and unfolding. And then it's released, and then there's 2 stems that span the whole bilayer between the cells, and then eventually a B barrel-like structure is formed, which allows for hemolysis to occur by this protein. And I also found in literature that different types of hemolysis, alpha, beta, and gamma, can all be expressed by hemolysin 3. And then finally, I found that the experimental protein structure was pretty similar to that of the literature. And then my hypothesis was basically just that Christia megaterium will exhibit hemolysis, whether it's alpha or beta, but that there will be some sort of zone of clearing on a blood agar plate. Experimental validation was that my bacteria did exhibit motility, which I did expect based on the genes. I was super happy because I did see hemolysis, although it wasn't complete beta hemolysis, it was alpha. It doesn't really show as well in this picture, but it was kind of like a brown color on the plate. There was lactose fermentation. Again, you can't really see it in the picture, but that was cool, and I did expect that in my predictions, and then didn't grow on the McConkey adder because it's gram-positive. And then for some more experimental validation, there was no antibiotic resistance, which I was pretty, surprised by, because there was genes for antibiotic resistance in the actual genome, as well as antimicrobial. There was genes for that as well. So I was surprised that I didn't see any antibiotic resistance or antimicrobial properties. That just may be because these are not the right antibiotics or bacteria to be testing my bacteria against. And then lastly, for the interpretation, I noted that my hypothesis was proven by my results. As we saw on the plate, there was alpha or partial hemolysis conducted by the bacteria, and that was supported by the genomic predictions in that hemolysin 3 can perform all 3 types of hemolysis. In the future, I would really like to experiment on human blood because that could have cool implications for medicine and developing different types of therapies, as well as how it lyses other types of cells besides blood cells, just to see if maybe it can be formed into some sort of other product. And then as well as visual observation of hemolysis due to the large nature of Christia megaterium, seeing if I could actually watch it under the microscope.
Now turning to my isolate, I had Chryssobacterium arthrosphaerae. I originally chose it because of its yellow color, and it was a bright yellow, and I saw when I isolated by itself on the agar plate, there was a lot of clumping, and there was almost a gel-like formation, probably a biofilm. Under the microscope and after a gram stain, I saw it was gram-negative, it was rod-shaped, and these cells kind of grouped together, and they were very small at this being at the highest magnification. Looking at the genes and the contig, there was 1 full contig of 5.8 megabase pairs, and there are a lot of genes relating to motility, hemolysin, antibiotic resistance, lactose fermentation, and antimicrobial potentials. So basically every experiment that we did during our labs. For our bioinformatic analysis, I decided to hone on the beta-lactamase antibiotic resistance gene that I was able to identify. I chose this because beta-lactamase is a very specific targeting, it's very specific to target antibiotics that are the beta-lactamine class. So its existence made it easy for me to choose a specific assay in antibiotic testing this. When we look at the subcellular location, we saw a lot of it at the outer membrane signals and extracellular, which makes sense because it needs to inhibit the beta-lactam before it actually affects the cell wall of the bacteria. Looking at the functional domains of my protein, which right here is the protein that was predicted by AlphaFold compared to what literature shows of very similar base pairs, very similar confidence. So we break this down to the domains. We have a penicillin binding protein dimerization, which is about the first 200 base pairs. And it's around this section right here. And that's what actually binds this beta-lactamase protein to the beta-lactam portion of the antibiotic. And then following is the penicillin binding protein portion. It says dimerization there. It's supposed to say the transmembrane peptase. That's what's actually cleaving the beta-lactam to destroy it. And that's this main portion right here, which you can see in both of the images. And that's a major portion of the protein, about 350 of those sequences. And then finally at the end, you see a disordered portion right there, just the final portion of the protein. So my hypothesis then relates to that gene in that if the C. arthrosphaerae is grown in the presence of beta-lactam antibiotics, it would still continue to grow via the production of beta-lactamases. Looking at some of the experiments that we did, I saw a tremendous amount of motility, which kind of surprised me because there weren't many flagellar genes, but I think it is due to the gliding genes that the protein had. Looking at hemolysis, it was difficult to determine if there was alpha, beta, or, well, not alpha, but beta or gamma because of the color of my isolate, the yellow color. But I ultimately determined that it was gamma, that there was no hemolysis. On Conkey agar, my bacteria was gram-negative, so it had the potential of showing the results on this agar, and with no with no lactose fermentation. Finally, to the antimicrobial assay, again, there were similar to the rest of my group's results. There was no antimicrobial potential. Turning to the antibiotic resistance, however, we see that both penicillin and streptomycin had growth, showing the antibiotic resistance of my bacteria. Vancomycin, it showed some resistance, and then we saw the beta-lactamase inhibitor. It showed some resistance, but also, again, it was affected in its growth potential. So my interpretation is that my hypothesis was correct because it survived against penicillin, a beta-lactam type antibiotic. And with this beta-lactamase inhibitor causing the bacteria to slow its growth shows that the beta-lactamase is most likely being produced for that survival. Follow-up experiments would include doing a knockout of that beta-lactamase gene and plating again to see if the bacteria would not be able to survive in penicillin type conditions. And it would also be interesting to see that if it would still survive when it's plated with streptomycin.
So finally, some conclusions. Overall, some major findings that there was definitely some success in our experimental testing. Cora and Shaylee's hemolysis genes were found to be functional and produce the phenotypes they were looking for. My antimicrobial activity was not found for my hypothesis, and Gray did find the antibiotic potential that he was looking for his hypothesis. Overall, none of our isolates had any antimicrobial properties across all of the tested species, and there was some antibiotic resistance for each of our isolates, a little bit at least. And all of our isolates were also modal as well, which is interesting to note. So looking more into the genotype-phenotype relationship, we learned a lot definitely throughout the entire semester, but that the relationship is not always quite clear-cut. It's important to look at various different elements to get a good idea of the relationship. Testing might reveal that the relationship might not exist as we predicted, as we all kind of learned today, and that the environmental aspect is a really large factor in sort of contributing to the phenotype, and so it's more than just the genotype as well, and that's a very important factor to consider. In the future, definitely more testing in the genotype space will definitely help, but there's so many tools and pieces of information that we can use already, which is really cool.
So to conclude with the significance is that we originally isolated our bacteria from a microbe-rich area like a compost bin, but the rest of us did as well, in different areas of soil. And so this is very important because soil bacteria can be studied for clinical and pathogenical mechanisms, not just those relating to soil and to the environment that they are in, seeing the antibiotic potentials that we saw in our bacteria. We also learned that it's very important that the genotype and the environment affect phenotypical expressions, and just because of the existence of a genotype does not necessarily mean that we will see our bacteria survive in antibiotic conditions or be able to fight different kinds of microbes. So at the end of the day, it's crucial to cultivate that 95% of underestimated bacteria, to learn more about them, learn more about the genes and the phenotypes that they can express, and see how that relates to different aspects, clinical, pathogenic, or simply environmental. There's our references, and thank you.
I did have a question. For all of you, like, what kind of suggestions do you think you could give to the community garden regarding like what types of compost to be putting in there, knowing what you know now?
I think like as wide of a variety as possible is great. Like the reason we selected the compost was because there would be the potential for not only microbes from all around campus, but like the interactions between the microbes. So maybe like expanding the variety of different types of like food waste or yard waste or whatever, but also like varying throughout time. Maybe introducing this as like a consistent project, and you know, testing community garden compost throughout different like additions of different types of things. I mean, that could be really cool, and seeing if like different, you know, microbes develop because of that.
Yeah, to add to that, it's like when we isolate our bacteria, it was in the summer, August, and that time, and so we might have had a different kind of food composition that promoted a different type of bacterial selection. It's also important to note that the bacteria that we had weren't really of the major 16S population. If I can get there. So I don't think any of ours were of the repeated family names, which are more important to breaking down those organic molecules. And so it's interesting to see that the ones that we isolated weren't exactly the ones that represented the whole community. And so even though we have some conclusions about antibiotic potentials and other motility, it would be more important, I think, to also focus on creating conditions to isolate these bacteria and do different kind of tests on them to understand what would benefit the community garden more.
So I know that we have the same bacteria, the Priastia like megatherium, and I thought it was interesting because when I was doing like my results, I only had like 2, I think, genes for like the homolysis, and then mine ended up resulting with a gamma.
Oh wow,
Instead of an alpha. So why do you think that there was like a difference?
I mean, it definitely could be because there's like different interactions between our different environments. Like I talked about with the breaking down of compost, all the bacteria have to work together to digest like the food and scraps and form it into a soil. So there might be more emphasis on hemolysis, not in the context of like breaking down red blood cells, but in the context of like lysing other cells. So that just might be more present in like our sample.
Yeah.
I had a question for both of you. This bee barrel, can you show me one of those images again?
I don't have any images of it.
Well,
I have like one
Yeah,
that
this one.
showed up in my.
We also had different, like I had
that's
hemolysis
good.
3,
That's good.
and you had HBO.
I had
so there's different
HBO.
hemolysis genes.
No, that's all. This good. So what membrane is that they're showing there?
It's probably the membrane of a gram-positive bacteria. It's allowed.
I think that's the membrane of the target cell.
Okay.
Well,
The target cell you're lysing.
I guess like there should be 2 membranes shown.
Hmm? I think that is the top of that slide is
Sure. So
is
this
the
would
outside
be
of
the-
cells. It's this like water or something. It's the extracellular environment, and that the hemolysin has come over and integrated into the membrane of the cell it's going to lyse. So that's good to sort of orient that. You know, which membrane is that? Is it bacterial or is it eukaryote? Is it the inside? Is it the outside?
Yes, it would be.
And then someone talked about bee barrel, said that word.
I think those.
what part of that is the B barrel? What is, or beta barrel? Go back to the other slide. Which part?
I had said that there was a barrel made of alpha helices.
The beta barrels are called that for a reason, right? In chemistry, you learn about two different secondary structures.
Yes.
And what are they?
The alpha helices
Or the
beta sheets.
beta sheets.
Yeah.
So what part of that is beta sheet?
The bottom part that's like.
Yeah, that's a beta barrel.
Yeah.
That red thing is the beta barrel. It literally forms a barrel, right? That's the thing that's literally forming a hole, a tube through the membrane. That's probably causing that cell to die. So when you say like B barrel or something, that's what it ultimately forms. And sometimes the components themselves, right, that's many proteins that make that up, not just one. So they don't necessarily fold into that conformation until they get integrated into the membrane too. I mean, it's really amazing cell biology, how that actually works.
That's why it's important to show this image instead of just a single protein.
Right.
Because allow the actual function, there's only function when there's like the actual structure.
It's actually showing you, right, that there's a monomer is number one. That's one of the proteins. Then there's like 1, 2, 3, 4, 5, and they form together. They all come together and make the actual multi-protein complex that causes the cell lysis. One of those proteins won't do it.
Yeah, I think that the binding component specifically helps like the L2 and L1, like those are called like the lytic components. So once like those especially come together, it like allows that for the like pool formation.
Yeah, it's cool, right?
It's really cool.
Forms, and then you imagine the red part sort of going down and poking a hole.
I have a question for Cora.
Sure.
So when I looked at some of the analytic activities, like the controls, I was always confused about alpha, because the alpha control we had, I don't think was a good control.
Right.
So I guess you didn't show your control results. But
No, I didn't go, honestly, I didn't go based on the control results, I went based on literature, because I know that in literature, beta hemolysis would cause a blood agar plate to almost show like a regular agar plate, like yellow, like how theirs kind of was, and then beta makes it like a green-brown color because the hemoglobin is like reduced basically, and then the gamma, there's like, it's just red. So I saw the green-brown color. It wasn't, it was in the middle of like what it would look like if it was gamma or beta. So
Gamma or alpha.
That was
Gamma or
why I
alpha.
interpreted it. As.
Remember, beta's better. Beta's full.
Yeah.
So it should be
was in the middle
between-
of- sorry, it was in the middle of gamma and beta. So mine was alpha.
so like.
Because mine was partial.
Yeah.
And I didn't have that like zone of inhibition because it was just reducing those hemoglobins within the cells that it was plated on top of.
Yeah. Like, where I'm trying to get to is, if you send this to a journal,
Right.
and say, I see green,
Right.
therefore
right,
it's alpha, they'll ask you, do this and that experiments to provide more proof that it is an alpha.
Like plating a better control for the alpha.
Very control, or think about other ways that you can test
That it's
if
alpha.
something has an alpha in your report.
Right. I think we're over time.
Great. Thanks.